So my talk is titled Deconstructing Epileptic Circuits in the Mouse Model of an SLC61 related disorder. And we are based in Glasgow Institute's UCSF. Our main questions in the lab are, how is the mutation SLC6A1 affecting the brain activity? What brain regions and cells are affected the most? And which cells are sufficient to cure SRD? So what causes epilepsy in SRD? So we were fortunate enough to work with Dr. Goodspeed in the natural history study to try to figure out and look at the epileptic activity in the brains of the patients with SLC6A1. So here you can look at a simplified montage of the EEG recording that was happening in a patient with S295L mutation. And a simplified montage in here, you can see that there are these high amplitude spikes in A and in B. And in A, they're starting on the two bottom traces of the recordings and kind of parietal and occipital activity. And this is called OIRDA, occipital intermittent rhythmic delta activity. And on the bottom four traces, you can see it kind of starts at the front of the brain and moves back to the brain. And it's called frontal intermittent rhythmic activity, delta activity that spreads to the OIRDA, to the occipital one. So this kind of slowing is very common in our patients with SLC6A1 mutation. Interestingly, these features are associated with absence seizures. So the S295L patient was diagnosed with absence seizures based on the slowing of the delta activity. So what happens in mouse models, preclinical mouse models with SLC6A1 mutation? And when talking about preclinical mouse models, we're talking about mouse survival, we're talking about epileptic activity, behavioral abnormalities, and then the main question that we want to answer is, are specific brain regions more susceptible to epileptic activity in SLC6A1 associated disorder? So I'll start with the first one. When we looked at the survival of these mice in our lab, we found out that when looking at the knock-in mice, the S295L mice, compared to the wild-type littermate controls, we did not see any changes in their survival. So they develop and survive embryonically normally, and they have approximately normal survival compared to their wild-type littermate controls. And they're sex matched, which means that both males and females are born normally. We call this Mendelian ratio to be normal. However, in the classic heterozygote and the knockout model that has been studied before the S295L model was designed, we found that the Mendelian ratios are skewed, especially in the knockout mice. You can see for every for example, 50 wild-type mice, the littermate controls that are born, there is a decrease of how many knockout mice are born. So these animals just do not survive normally, and they are probably embryonically lethal, and therefore we believe that this animal model is probably too harsh of a model and does not really depict of what we're trying to model in our preclinical animal studies. However, the sex ratio was also maintained. So in here we say that the knockout Mendelian ratios are skewed and the knock-in Mendelian ratios are normal, making it the S295L model quite an efficient one to study these types of questions that we were trying to answer. The next one, we were asking about the epileptic activity. So in here, what you can see is we performed the EEG and EMG recordings of these S295L mice. And on top, you can see the muscle activity of the mouse. And the mouse is moving normally. And then there is this behavioral arrest or a stop in the activity of the mouse. And on the bottom, you can see what's happening, the high amplitude spike and wave discharges. And this is a type of a seizure. And we call these seizures absence seizures that are correlated with the behavioral arrest in these mice. And when quantified, you can see that the seizure count is quite increased in these animal models compared to the littermate controls. What about the heterozygote mouse when just one allele is completely lost? So we do see this similar kind of activity in a hat mouse. We see a behavioral arrest that is associated with a spike and wave seizure discharge in these animal models. And also quantified here on the right. You can see that there are 3 groups, and the third bar is the knockout bar. And you can see that the full knockout is actually showing so much epileptic activity that that's not even close to what the heterozygote or S295L is. So the knockout is a very robust seizure model. Again, it's probably not really modeling what we're seeing in our patients. However, it might be also useful for the triad of some of the therapeutic approaches as well. So moving on, we wanted to do some pharmacology studies in these mice, and these are classic pharmacology to test of what these epileptic seizures are responsive to. And the first one we studied was the NO711. So the NO711 is actually blocking the GAT1 function, so the protein the SLC6A1 encodes for. So in here, when we record during the day, you can see that the middle bar, I'm trying to see if I can show it with this stuff. I'm not sure if I have a pointer. Oh, here you go. So the NO711 in the middle bar, it increases the epileptic activity in the wild-type mice, meaning that when you block the activity of GAT1, so in here, in the wild-type mice, we're technically making an animal model of a loss of function of GAT1 using this drug. But when we treat the animal with ethosuximide, and this is an anti-seizure medication that is usually used for the for absence seizures in clinic, it was able to completely abolish all of these seizures activities from happening. Interestingly, when we look at the S295 animal model, we see that at the beginning, they already have the baseline activity, but when we treat them with the NO711, when we block the leftover GAT1 that's leftover from the normal allele, we can further increase the seizure activity, meaning that leftover GAT1 is responsive to this exacerbating drug. However, when treated with ethosuximide, again, it was fully abolished from this activity. We have done similar pharmacology studies with the knockout line. In here, we have done the same with the wild-type mice, the hat mice, and the knockout mice. Interestingly, the hats showed the exactly same activity with the NO711 and ethosuximide as our S295L model. In the knockout mice, however, we did not see any exacerbation by the NO711, and it was kind of expected. There was no target for NO711 to act on. Thus, we could not make the phenotype even worse. And ethosuximide, unfortunately, was not able to have an effect on these seizures as well. So the next question we wanted to ask was, does the state of the mouse affect the epilepsy? So we looked at the active state mice and the quiet state, likely to be in sleep. And what we see, comparing to the wild-type mice here on the left, during active state, we have some of these seizures that are associated with the behavioral arrest. But take a look at the quiet state. There's a lot more of this activity in the quiet state that is happening in here in the S295L model. So this is something that we're working on and trying to really carefully characterize the sleep in these mice and also look at the activity, epileptic activity during sleep as well. So the next question is about the behavioral abnormalities in these animal models. So we know that the behavioral phenotyping is an essential part of both basic and translational neuroscience, and there are a lot of mouse behaviors that can be quantified. We have inactive behaviors, we have exploratory, maintenance, agonistic, affiliative, sexual, and maternal behaviors. So behavioral neuroscience really focuses on trying to identify which ones of these evolutionary conserved behaviors we can study for clinical animal models to then that we can target with to see whether we can model some of the behaviors that we see in our patients, but as well whether we can fix these behavior alterations in our preclinical animal models. So what have we done with our S295L mice? We checked multiple neurobehavioral domains in locomotion, anxiety, cognition and memory, sociability, muscle strength, and nesting. And what we see is that locomotion is decreased in multiple tests, including open field, light dark box, white maze, and rotarod, and the muscle strength is also decreased on the grip strength. And I'm going to show you a couple of examples of those. So, for example, here in the open field activity, what we see is in females, these are the tracks of how they are running in the open field. The tracks are decreased. There's less running in this female square for the S295L model. And when quantified, if you run this behavior together and you're not powered enough to identify or stratify for the different sexes, you might be able to miss this phenotype. But in here, if you stratify it into females and males, you can see that there's a reduction in the activity that is happening in females. Similarly, when looking at the grip strength, what we see that overall when sexes combined, we've seen this activity, but this phenotype was really driven by the females as well and not in the males. And another locomotion and somewhat muscle strength testing is the behavior assay called rotarod. When these are moving, this is a moving rod, and animals are trying to catch on and retain their position on the accelerating rod that is moving. And when combined with both sexes, we do not see a difference. However, the females are showing a reduction that males do not show. And you can see that there's a lot more ends in here that I run, and this is a repetition cohort in completely separate cohort to verify whether I see locomotion differences from the preliminary cohort that we have done. So overall, we see a couple of behavioral phenotypes. However, they are quite mild. So to really address this question with the mild phenotypes, researchers have moved forward and tried to really identify ways to analyze behavior using artificial intelligence and machine learning. So not only SLC6A1, but other epilepsy and autistic disorders have problems of identifying behavioral phenotypes very well. So this particular one is called behavioral syllable sequencing. And what is done in here is that as an animal naturally behaves, it goes through a sequence of different behaviors. And for example, in here as an orange bar, an animal can rear up, and these are just points of the body as an animal is rearing up. And then an animal is pausing and then rearing down. And you can see that technically all of the behaviors that animals are going through is just a sequence of these syllables of the behaviors. And this paper has been recently published by the Soltis group from Stanford. They're our collaborators, and we have published a commentary about this method to be used in epilepsy models as well. So what I'm doing in here is this is an animal model. This is an S295L mouse. This is just running in the open field, and I'm recording the animal on the bottom. And you can see I'm tracking all of their positions of the paws and the tail, and just trying to identify what an animal is doing in an open field, kind of unbiasedly using machine learning approaches to identify what an animal is doing without any of the classical preconceptions of what I'm trying to identify. And what I get from here is this plot of every data point of the animal body parts positioning in space. And from there, what I can do is I can organize these behaviors in these syllables, what I showed you on the previous slide. And these are technically the behaviors. This is one of the behaviors that an animal is performing. So what is this behavior? So you can see this is a syllable. It was by machine learning called syllable number 20. And if you look at it, this is a movement forward. So this is a very unbiased approach to say what is happening in locomotion using this syllable. And then here you can see when I verify, this is an animal actually moving forward. So this is trying to understand animal behavior from a very naturalistic way and figuring out whether us as scientists not looking at animals and trying to tease out different behaviors that what we're used to see, whether we can use and analyze their natural behavior that we might not think of as important to be used for characterization of SLC6A1A models. So interestingly, the Dravet animal model that Soltis Lab has worked with, they have never been able to identify behavioral phenotypes in this Dravet model. However, using this approach and identifying these naturalistic behaviors, you can see that they found, comparing to their littermate controls, this Dravet animal model was found to have differences in these specific naturalistic behaviors that they now are showing are good outcome measures to use for testing different anti-seizure medications, but also identifying whether there are any side effects from the seizure medications that can show up on these behavioral outcomes. So we're excited. The analysis is ongoing on these mice. So I recorded all of the open field naturalistic activity for these animal models, and stay tuned for the results of it. So the next question we're trying to answer is, are specific brain regions more susceptible to epileptic activity in SRD? And how we're trying to answer that question is we designed these devices, custom-made devices, where we're targeting different brain regions to identify what kind of activity is happening in multiple brain regions. So when we analyze this behavior, we can record from EEG or local field potentials for multiple brain regions. And you can see I'm targeting the cortex, motor cortex, somatosensory cortex, local field potential from L4 and L5 of the cortex, hippocampal activity, and the thalamic activity. And bottom line in here, all activity is normal. However, right before the REM sleep happens, we see the seizure happening in the M1 cortex that is coinciding with something happening in the thalamic LFP. And when we look closely on the thalamus, when I enlarge the first spike that is happening in the seizure, what we can see right preceding this first spike in the seizure, we can see the bursting activity in the thalamus. So thalamus is a very important brain region that controls absence seizures, both in humans and in preclinical animal models of epilepsies. And then here we have first evidence that thalamus is involved in the seizure expression in these mice as well. So Jean's work in her postdoc published in Nature and shows that when targeting optogenetically thalamus and the interconnected cortical region, you can identify the signature of the thalamus that will precipitate out in the epileptic seizure in the cortex that we usually detect with EEG, and you can prevent that seizure from happening when you shine light onto the thalamic neurons that express the optogenetic marker. So this is kind of in hope to develop the closed loop stimulation protocol in our mice as well, to see whether we can stop these seizures before they are even happening. An interesting thing about the thalamus and the EEGs that we are analyzing, we're seeing that some of the seizures that are happening here in M1, they seem to disrupt the sleep that is happening here in EMG. So when an animal is sleeping and then when a seizure happens, an animal wakes up. So we think that some of these seizure activity might be also contributing to the sleep disruption in these mice as well. Interestingly, when looking at the in the S295L animal model, comparing to the NREM sleep, which is one of the sleep phases, comparing to the REM sleep, our seizure activity in these animals seems to be much more robust during the REM sleep rather than NREM sleep. And this is something that we're trying to analyze as well and trying to catch as many of the REM events in these animals as well. So moving forward, we're doing a lot of the thalamic targeting for the SLC6A1 gene therapy. And in here, as a proof of concept, we're showing SLC6A1 expression in the thalamus already. And then here we are targeting them and with this specific AAV construct in the astrocytes to see how far it spreads and what's the efficacy of our AAV treatment as well. So where is SLC6A1 expressed in the thalamus? So I showed you that we have expressed them in the astrocytes because in the thalamus, using this RNA sequencing, we see that there's a lot of SLC6A1s, particularly expressed in astrocytes at a much higher expression levels comparing to even the GABAergic neurons. So it'd be very interesting to see whether targeting astrocytes rather than GABAergic neurons specifically would be a great therapeutic target for the viral vectors. So the collaborative project that we're working with the Nicholas Trish lab and the JAX labs, is working with the new animal models. So I talked to you about the S295L and the knockout models. This new animal model is SLC61 flox animal model. So this is technically a wild type animal, but when you add the Cree, you create a knockout. And this is able to answer the question, is cell-specific SLC61 necessary and sufficient to cause SRD? And now with Jennifer San Miguel at JAX Labs, which she's going to be talking today as well, we're working in characterizing this new animal model called SLC61 stop flox, which is technically a knockout, but when adding Cree, it creates a wild type. Trying to answer the question, is cell-specific SLC61 necessary and sufficient to cure SRD? So these are complementary mouse models trying to answer these very specific region and cell type specific questions. And with that, I'll conclude that SRD mice have absence epilepsy. Mice have normal embryonic and early postnatal survival. They have sex-dependent locomotion and muscle tone abnormalities. And the thalamocortical bursting synchronizes with epileptic activity detected in the cortex. And the pilot data suggests seizures can disrupt sleep, but future directions will determine whether that's the case or not. And with that, I want to thank our lab trying to answer these questions with the goal to cure the SLC61 related disorder. And next steps, we want to try a lot of pharmacology approaches, gene therapy approaches, and the ASO approaches, now having a great platform of multiple animal models that we can test them in. Thank you to Amber Freed, all of our collaborators, both internal and external, our funding sources, especially SLC61 Connect, and NIH. Thank you.